Hello friends, in the previous video we discussed about horizontal gene transfer in bacteria. We have discussed that there are three methods of horizontal gene transfer in bacteria: transformation, transduction and conjugation. In this video we are going to discuss about bacterial transformation. What is bacterial transformation? It is an active process of horizontal gene transfer by which some bacteria take up foreign genetic material, that is naked DNA, from the environment. Active process means it requires energy. It is ATP dependent process. It was first discovered by Griffith in 1928 in Streptococcus pneumoniae. Avery, MacLeod and McCarthy in 1944 demonstrated that DNA is the transforming factor. These we have already discussed in detail in DNA as hereditary material video. You can refer my video, the link is given in the description box. So now the question arises, how the free DNA is present in the environment. When any bacterial cell dies, it is subsequently lysed and DNA is released. These are taken up by the bacteria, recipient bacteria present in the environment. How? Recipient bacteria take up free DNA by the ability known as competence. Such cells are called competent cells. What is competence? It is ability of recipient bacterium to take up free DNA and get transformed. This competence is inheritable characteristics in certain bacterial strains. Some species are naturally competent. Competent bacteria encode proteins, receptor proteins known as competence factors, which facilitate binding of DNA fragments to cell surface and its uptake into cytoplasm. Examples: Streptococcus pneumoniae, Bacillus subtilis, etc. Transformation does not occur naturally in all species and species in which it occurs naturally. It does not occur in entire population but only in competent cells. Now let us see the mechanism of transformation or the steps involved in transformation. The entire process of transformation is divided into following stages. First of all, reversible binding of double-stranded DNA fragment takes place. This takes place by the binding of this double-stranded DNA fragment to the competent factor or the protein present on the surface known as the receptor protein. Then the double-stranded DNA is converted into single-stranded DNA molecules by nucleolytic degradation of one strand by the nuclease enzyme. Then irreversible uptake of donor DNA takes place. This single strand of the donor DNA is taken inside the cell into the cytoplasm. Within the cell, single-strand DNA is bound by single-strand binding protein in order to protect it from nuclease enzyme. Then the integration of single-strand donor DNA takes place into the recipient chromosome, and this integration occurs by homologous recombination. At last, segregation and phenotypic expression of integrated donor gene or genes takes place in the recombinant cell or transformed cell. Let us understand this with the help of the figure. These are the free DNA fragments or the donor DNA fragments present in the environment. Suppose this fragment with the gene A+ comes and binds to the surface protein of the recipient bacteria. One of the strands of the donor DNA is degraded and the single strand of the donor DNA is taken inside or pulled inside the cell. Single strand binding protein comes and binds with this single stranded DNA in order to protect it from nuclease enzyme. Then the integration of the single stranded donor DNA takes place. This segment of the recipient DNA is replaced and single-stranded donor DNA is integrated. This strand which is replaced is degraded. Now it is known as heterologous chromosome because there is mismatch present. One has A allele and the other has A plus allele. Now replication of the DNA takes place. The two strands separate. This strand serves as the template for the synthesis of the new strand which has allele A. And this strand which has A plus allele serves as the template. New strand is synthesized which also has allele A plus. Then the division of the bacteria takes place. Now two bacterial cells are obtained. One has A allele as the parental cell. So it is same as the parental cell. But the other cell has A plus allele. So this cell has the recombinant DNA. So this cell is known as the transformed cell or the recombinant cell. Now let us see some of the facts related to transformation. We have seen that transformation is active process. When the DNA, donor DNA is pulled inside or taken inside, then the energy is required. This energy is derived from the degradation of the complementary strand. During integration of the single-stranded donor DNA fragment, a segment of one strand of the recipient chromosome is replaced. So, as discussed earlier, this strand is integrated by replacing the fragment of this strand of the recipient DNA. Donor DNA fragment is known as exogenote. Recipient's genetic material is known as endogenote. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.